A very good morning to all of you. So today we are having a special seminar from Dr. KB Saxena, who has been working at ICRISAT for about 40 years. And so today is your superannuation day, we call it like that? Or? Yes. Okay. So Dr. Saxena is superannuating today on the 31st January 2014. And so we thought that let's have a seminar from him where he can summarize his work during last 40 years, his experience working with a range of people with on different topics, etc. And of course, finally some words of wisdom from Dr. Saxena. Sure. So we thought that yeah, then we should have, and this is, in fact earlier this was really very difficult for us to organize the seminar and farewell because many of senior leaders from ICRISAT they are away, because even I myself was also there. We have right now for last 2 weeks we have been organizing the meetings, regional planning meeting in Western Central Africa, then we had in Eastern Southern Africa. In fact I was supposed to be in Ethiopia today as well, but we thought when Dr. Saxena is giving seminar we need to be here. So then we have organized seminar today, and then in afternoon we will be having farewell. So we will not be talking about farewell and good words or tears, etc. right now. We will be doing this thing in afternoon. Right now we will be having this scientific presentation. And we have requested Dilip and Pradip Modi and their team for recording this presentation. So because several of our colleagues from African location, they may not be able to view this presentation live right now, but once we have the recording, then this recorded presentation can be shared by anyone else. So again, so welcome to all of you in this presentation, and now I would like to request Dr. Saxena to share his work. So please welcome once again Dr. Saxena.
Thank you. Good morning to everybody, especially my friends in Australia, Dr. Chauhan and Dr. Rao, who have confirmed that they will definitely like to listen my seminar, and also to my friends in Delhi and Gujarat, who have also confirmed they would like to listen what I say here. So I will be very careful what I talk. Okay, so no nonsense talk. And also my friends in Africa, whether they are able to listen or not today. And as Dr. Rajiv has just mentioned, he will provide this material to others to listen. Listen what I say here. And at the outset, I would like to thank Rajiv. Really very much thank you very much, Rajiv, because you could make it from Africa to India. Very long journey, I understand. And this shows his commitment towards Pigeonpea. And I personally appreciate it. And I hope this commitment will continue for next 2 decades until he retires. So, before moving to my seminar, I would like to draw your attention on this slide which I should put on this screen. This is the latest press release from ICRISAT which has appeared in Times of India yesterday. So we thought we'll take it here. And it's very inspiring. Inspiring in the sense that now people are knowing about the technology, what ICRISAT has done, and why it is so important, particularly in Pigeonpea. Hybrids have been developed in many crops, but in Pigeonpea, we are not able to do it for the last, I can say, 1 century. The yields are almost stable around 700 hectares. I don't want to show that slide because that is very depressing slide. Yield is straight line all the time. So we thought we will move forward with hybrid technology, and this particular news is based on our field meeting here which we organized. We called our seed producers and discussed with them and all that. And most of the people said that seed production in hybrid will not be a problem because they know the trick now how to produce seed, and that is how, if seed is produced, yields are high, that means no problems in taking technology away from us. So we are at that stage, and I'm sure ICRISAT management will also support us in pushing the technology. I'll talk more detail about it a little later. Now, Dr. Rajiv had mentioned what I would like to talk about what happened in Pigeonpea 4 decades, because many of you must have not born that time when the Pigeonpea program started. So it's a good beginning at least to share quickly what happened, how good we were at one time and how bad we were at one time. and then how we came out of all this trouble. So this was our vision in 1974 set for Pigeonpea. As I mentioned, productivity was 700 kg per hectare, and still it is same. And you can imagine how ineffective our breeding programs were, that we could not increase yield in spite of so much effort being given to improve the productivity of this crop, not only in ICRISAT but in national program as well, but we did not succeed. Of course, we got success in increasing area, and that was because of the factors such as incorporation of disease resistance in high-yielding varieties, development of early varieties, and greater awareness. So area increased, but the productivity still remained the same, and that was concern to all of us, not only researchers but management also and government also, how to increase it. So we started putting our eye on that we must increase productivity somehow. Then at that time in 1974, awareness about Pigeonpea was very, very little. In fact, in India, many people didn't know what is Pigeonpea, because in India we call it red gram. But once we started communicating with them through many media and meetings and our travels, now things were quite improving. And another target we set in 1974 was to develop important scientific information, because that was lacking. And scientific information is always the base to put a step ahead. And also cropping system, because cropping system also very important in this crop. And then how we should do this. This was also problem. I'm not going into detail of that. We can discuss anytime we want. But after a couple of years, we came out with the fact that at least we know some reasons why it is very difficult to improve this crop. There are basic difficulties, such like perenniality. So that means this plant does not die, like sorghum, like wheat, like maize, and it remains alive. And that is not good for high yielding. It is good for stability, but not for high yielding, because plant does not transport all the food material to the reproductive parts, and it saves a lot of things for the bad days. So that's why that was the first problem we identified. Until we discover an annual type, perhaps yield per plant or per unit area, very difficult to improve. And still today, we are on the same place that we could not find any true annual type in this crop. So thinking is how to go ahead with this. Now, other problem was photosensitivity. That means this plant can flower only in short days. It will not flower in longer days, and that means that is restricting our production system. So this particular thing we addressed very well by developing early maturing variety, and we demonstrated that early maturity was linked to photoinsensitivity. That means we could grow these very early types in different planting dates and different places, even up to 46° north, where people didn't really think that we can grow Pigeonpea ever, but it wasn't possible. Now, other point was we had that how we can use the right maturity we have in this crop. The maturity for this is really wide. We have plants, we have Germplasm now, which matures in 80 days, and another one, other extreme, more than 300 days. And each maturity group is important. We cannot say this is good or this is bad, because each has its own production system and its production area. So that way, we decided that we will stratify our breeding program accordingly. And in that context, we established 2 centers in India. One was in Hisar to breed early maturing varieties, and another was in Gwalior, where we breed long duration varieties. And at Patancheru center, we will breed medium duration varieties. So that's how our breeding program was stratified. And I'm very happy to mention that this was a very successful move from ICRISAT management. We got very few varieties released from Hisar program, early varieties, and also long duration varieties from Gwalior program, and of course medium duration. I think still those varieties are ruling. Now, major objective we started that time, that first we should know what our germplasm is. Until we know that, we cannot use germplasm for any good purpose. So we put a lot of resources. ICRISAT put a lot of resources to characterize germplasm for various qualitative and quantitative traits, including disease resistance and other traits. And they came out with a catalog that was very useful, not only for ICRISAT but globally also, so that people could select material what they want for their area. And the very big program which I started was breeding varieties. And I was given responsibility to start with the biggest ever possible dial made, 28 by 28 dial. You can imagine those breeders sitting here, they can imagine how difficult it is even 7 by 7 dialer. But we completed 28 by 28 dialer. The consequence of that was that in 2 years we had plenty of breeding material in our hand. And that material was shared with many people in North, including India and the West Indies and Africa, wherever they wanted. So this material, the variety developed from this material are still doing very well. Now, that time, we also started thinking that why not we exploit hybrid breeding in this crop, because we have around 30% natural outcrossing. So this idea was not mine. It was the idea from Dr. John Green, late John Green, who was cotton breeder in US in a private company and joined ICRISAT Pigeonpea breeding program. So he thought, let us try. That time many people discouraged us. Hybrids will not work in any pulse crop. But we thought, being an international institute, we must try new things, then only things will work for future. And we started working on that under his leadership. And the first step was to look for South Atlantic male strategy. And we succeeded very late in that. And then other objective was to find out ways and means how we can encounter various biotic and abiotic stresses, especially biotic stresses, that diseases and insects. And then also we had a very big program on human resource development. We used to organize training program, short-term training program, medium-term training program, long-term training program, and most of the cases were funded by ICRISAT, because at that time, right in this building, in this wing, we had a huge training program, and they had a lot of money to support training from different continents. Now, briefly going to that, what we achieved, because we can't go very much in detail. As I mentioned, germplasm was successfully classified. And we developed a lot of scientific information on that, which I will talk a little bit on that. And various varieties were released, which are still popular. And genetic male sterility was identified, and we started working on that. And very important thing was, which our leader, Dr. Green, always emphasized, we should develop our output activity based on interdisciplinary approach. That means breeders will not have their own program. Breeders will have program with pathologists and entomologists and all physiologists, especially physiologists are very close to us. So we developed that kind of program. So we used to have planning meeting together, small group then big group. So always we were thinking, we were knowing who is doing what and why. That was important. If you are doing something, then you should know what my colleagues are doing, why they are doing, and what is the consequence of that. And that time, another good thing was that money was not a problem. Nobody talked of money. We simply talked of outputs only. And then we selected two very good materials. One was a clistogamous material that had a very unique characteristic that outcrossing was less than 1%. That means what? That means that we have converted partially outcrop species into totally self-pollinated, like wheat and other things, right? And that material is still here. We are transferring that trait, very simple to transfer because it's single recessive gene. And also we developed a high protein program that was closed in 1996, but we could develop lines with 32% protein. You will not believe it, but it's true. Normal Pigeonpea is around 22% maximum or 23%, but we could develop lines with 32% protein, and this character was transferred from wild relatives of Pigeonpea. I personally was handling it, and we used to get very good collaboration from biochemistry department, because we used to analyze thousands of samples every year. So these are some champion varieties which I have outlined here. ICPH 88339, and this is very unique how we developed it, and my friend Dr. Y.S. Chauhan was very much involved in the breeding of this variety with us. So this was a very good example how breeders and physiologists can get together. And this variety has less insect attack according to what data he collected. I didn't want to contest that kind of statement. So, but this variety is still very popular. In fact, our two major programs in hills and in Rajasthan are based on this variety only, and also this variety being grown in Philippines on a large scale now to introduce in a rice system. And then 7035, another fantastic variety. We don't take credit for breeding this variety because this variety was a collection from Madhya Pradesh, and is a really gem. It is resistant to wilt, all races of wilt, so far we know, all established that we know, high sugar, 8% sugar in this, and good vegetable type. So this is still popular, and this was the first variety released outside India from ICRISAT material. That was in Fiji. In Fiji they released it as Kamika, so still we call it Kamika. Now, the major breakthrough came when we really released our first hybrid, Pigeonpea hybrid, that was ICPH 8. It was very difficult to convince ICR because they were our partner also, but the DG of ICR always said, no, test more. So normal variety is tested for 3 years in ICR program. This variety was tested for 5 years. And then we were asked to demonstrate this in on-farm trial in Madhya Pradesh and in Andhra Pradesh, all peninsular India, and Maharashtra also. So we demonstrated in farmers' field on a very large scale this hybrid is good and it has a real head process which can be exploited. So this hybrid was finally released by ICR with ICRISAT in 1992. Now, quickly passing on to what kind of basic and very useful information we developed. Number one was the phenology. Phenology means how plant growth and spread and all those things. That varies from planting date. And this was because of the photosensitivity and temperature sensitivity of the crop. That means that we have to be very careful in breeding varieties from different areas. We cannot say all the varieties developed at ICRISAT will perform well everywhere. No, that's not possible. The sensitivity was very strong, so we had to be very careful. Now, second point, the relationship between seed size and yield. Everybody wants big seed, right? And we struggle all the time. How big seed can be carried away with the high yield, but it's not possible, and we found that relationship is curvilinear. Curvilinear means it will like this, So initially up to 10 grams we can increase yield and seed size, and after 14 grams if you increase seed size, yield will come down, and there is a free zone 10 to 14 grams. So we settled down there that all the variety released from ICRISAT will have that kind of seed size plus disease resistance always. Now, G into E, always important in all the crops, nothing new about it, but in Pigeonpea it was very, very strange. And we put our heads together and we still could not solve the mystery that why the variance of F2 is similar to variance of inbred lines. Those who know the Pigeonpea or those who know plant breeding, they understand this is totally against any breeding law. It cannot happen, right? The variability in F2 is maximum theoretically, and inbred line should have minimum variability, right? But for yield, we found is strange. And Dr. Gurin always on my head, KB, this is not correct. And finally he was upset, so upset that he give me data, I will calculate. So when weekend he took all the data from me and calculated, and one day he comes and says, KB, you are right. This is strange. That means very high implication of this is that we cannot do single plant selection for yield in F2 generation. Right, this is very, very important implication of that, and we avoided that. We started selecting for in the bulk progeny rows rather than going for single plant. So that was very important thing. Another thing we found that the gene action which is important for that, and we found that both additive and non-additive gene actions are involved with yield, and that is very important. See, additive gene action is very important for breeding pure line varieties, and non-additive is very important for breeding for hybrids. In hybrids, we can exploit, in fact, both additive plus non-additive, and that's what we are doing now. Now, other thing we found during that study that sink in this case is limited. Again, our physiology team, led by Dr. Johansson and Chauhan, they really demonstrated that it's not the source which is limiting in this crop. What is limiting is the sink. That material is not being transported to the pots, and lot of flower drop is there. So flower drop was a nightmare for most of the agronomists and and physiologists, but not to us. We knew that Pigeonpea produces a lot of flower, all flower cannot set pods. So that way we decided that how we should move on that. Then GMS hybrids cannot be commercialized. That was it. Maybe we knew it, it's not that we did not know it, but still we wanted to carry with GMS because we wanted to develop some basic information about hybrid program, and number one was whether heterosis is there or not in this crop. That's the last point you can see here, and we really demonstrated that plenty of heterosis means hybrid vigor. Plenty of hybrid vigor is available in the crop which can be exploited. And another important information we generated from that, whatever outcrossing we have, 25 to 30%, that is enough for hybrid seed production, and we really demonstrated that now in large scale at present. There is a lot of information generated on that. Now, the sad news, in 1996, those who were here, they know how we passed every day, because this is the time when retrenchment was at large. Many people were removed from ICRISAT because of financial problem, and most sufferer were Pigeonpea. And the argument was Pigeonpea 80% is grown in India, better remove it from the mandate crop. So decision was taken by management. It was announced that Pigeonpea is no more a mandate crop of ICRISAT, please pack up. So 80% staff of Pigeonpea breeding was removed that time, and they said, sorry, no funding, we can't do anything. So it was no problem, we took it very sportingly, but did not give up. I did not give it up. And then what happened? This is very, very important slide for hybrid breeding on for Pigeonpea. Right. What happened? We knew that we are at the verge of getting cytoplasmic male sterility, which nobody ever had in any legume crop, which could be utilized for hybrid breeding. And in one of the lines, and the field was near our lake here, I found that one progeny where 11 plants were grown, that is back cross progeny, all the 11 plants were male sterile. And I said, that's it. I went and talked to Don Byatt, who was our DDGR. He said, no way, KB, we can't do anything. I said, you can spend some time with us. He said, sorry, no time for you. You have no more stuff. So anyway, somehow I convinced because of my connections with him while I was also working in Australia with him. He said, okay, let us go to field. So next day we went to field, and I said, no, you don't do anything, just 11 plants you see. He was the breeder, John Byatt was the breeder. So he saw all the 11 plants, and you know what he said? Oh, and what next word he said, I can't tell you here. It is a CMS, right? And this was the words from him, and he was so excited himself that he said, okay, let's go and talk to DG. So we went to Dr. Ryan, he went to field, and then he decided, yes, very good, we'll retain Pigeonpea, but only hybrids. Two conditions were there. He said, we'll keep only hybrid, but no funding. We can give your salary only. I said, fine with it, no problem. So I approached Maiko. Dr. Barwale willingly gave $8,000 to me. I said, you manage with this, we will see later. So anyway, so it was all right. Now $8,000 has no value in ICRISAT, but that time my God, it was so good for us, news for us. And that was the first private sector funding. And when ICR heard of this, they started making a lot of noise. How ICRISAT can attract, it is public institution, how they can attract public, private funding. But somehow the matter was solved by our management, no problem. Now, after that, a second phase started. Second phase of hybrid Pigeonpea breeding. And that was the developing cytoplasmic based Pigeonpea hybrids. And after a lot of work, we could really develop a A4 CMS system. That means this male sterility is driven by cytoplasm of a cell. It's not driven by nuclear genes. So it is very difficult to breed cytoplasmic male sterile line, but it's very easy to use in breeding program. So it was worth waiting for about 30 years and getting this line. And then our group started working very vigorously to transfer this male sterility into different productive types. And then soon we started developing hybrids, and also simultaneously we continued working on seed production technology because that was the backbone of that. And also we involved a lot of NAS partners in this because we thought we cannot do it alone. So ICR granted a special project with about 9 universities and ICR center to work with ICRISAT on this, and we successfully really generated a lot of information on hybrid technology. Now we talk of hybrid. Now history is over. Now what happened? Why hybrids really? These are the benefits of the hybrid: more vigor and more yield. That's what we can see here. And another important thing was that the hybrid plants had about 40% more root mass. And this work was again done by Dr. Chauhan and Dr. Johansson here. And this is very good work where they really demonstrated why hybrids are so sustainable compared to normal varieties. And that's why it works. It also shows drought tolerance. Then also we really concluded that the hybrids can be grown very successfully under parocrop and intercrop also, and also under high input and low input. Both hybrid will perform better in all the agriculture situations. Now, hybrids were identified for release, and this was the first one. I called it brown gold because of some good reasons. So 2671 was the first CMS based commercial hybrid which was released in Madhya Pradesh with ICRISAT, and then 2740 that was released with all the efforts from Samir Kumar in Andhra Pradesh. And then we have 2 more hybrids which are very good in performance, 3762 and 3310. In fact, these 2 hybrids are in on-farm trial, and we are getting very good feedback on that. And any year this can be released for further use. Now, very quickly on the performance of hybrids, I don't want to go all this till. Please look into the last line. Hybrids are 46.5% better than varieties, and this was a very big gain. And this was, you can see the number of farmers who evaluated this. The data is from more than 2,000 farmers, and it's not easy to generate this data. And I really appreciate the effort of Vijay Kumar in really doing this, the distribution sheet, monitoring it, collecting data, interviewing farmer. Lot of things was done by my colleague Vijay Kumar, and this we generated very good data set on that. And based on them, the hybrid was released, and the same thing put in the form of graphs. Now this was the first demonstration in Andhra Pradesh, big demonstration, and that was organized by a farmer here in the middle, and he collected seed from National Seed Corporation, not from us. And when he cultivated his crop, he found it very good. Then he called Vijay Kumar, my God, we have very good crop here. And why he said because he got average yield of almost 3.5 tons per hectare, which was not really thoughtable in Pigeonpea, especially in Andhra Pradesh. This kind of yields are very high. And this farmer really became our ambassador in this area, and then he was later awarded Best Farmer Award by Andhra Pradesh government, especially in pulses. Now you can have a view how it looks. 2671, this is in Akola. Then you can see how it performed in Maharashtra, Osmanabad. Then you can see Ranchi, that is Jharkhand. You can see here the upper picture shows how hybrid is loaded with pod, and lower picture shows how the local variety Bahar does it. So there is no need of data set here. Just you can see yourself and decide which is good, which is bad. But it was important to collect data, and we had a lot of demonstration in Jharkhand also, and we found that hybrid was very, very high, like 69% better than varieties. All this data was collected by the university staff there. So we are quite excited. And now this year actually, the DAC has granted a special project to Jharkhand to pursue this hybrid. This is important. Now they have realized that without hybrid they cannot increase yield. So they asked them and to put up a project, and I took 2 days to write project for them. So now it is accepted there. So this kind of yield we got of the same hybrid in Madhya Pradesh where it was released, and you can again see what kind of gains we have. Again, it confirms what we got earlier. Yield advantage of 46%, which is very, very high as far as Pigeonpea is concerned. So this particular hybrid was released in Madhya Pradesh, and we got a lot of support from Dr. Tikle, who is a PNP pulse breeder in CO, and Mukesh, who is a PNP breeder in Indore. They really put a lot of efforts. Mukesh was involved in seed production technology, while Tikle was involved in this in the testing of the hybrid. So that way, jointly had a lot of input from all these people. Got it. Now, second hybrid. Second hybrid even better. The second hybrid was released in Andhra Pradesh in 2012, and these are the performance in Andhra Pradesh. Average yield 1.5 ton and productivity gain about 39%. So these are the kind of thing we were looking forward in our breeding program, and this is the again data on 2740 in 3 states, Madhya Pradesh, Maharashtra and Andhra Pradesh, and you can see the superiority of hybrid was again more than 40%. Now, coming back to production technology, I think this is the backbone of hybrid program. If we cannot produce seed economically, nobody will take, no seed producer will accept it, and your technology will go nothing except waste. So we have to work a lot of things on the seed production technology. And again, I would like to really appreciate the contribution of Vijay Kumar, who really put all efforts not only in developing this technology here and finally settled with 4:1, 4 female rows, 1 male row. And also he demonstrated in other areas also that whether this ratio is optimum or not, and then we found a very strange result that in different places the ratios had to be different, from 2:1 to 8:1 or 6:1 like that. It all depends on the availability of pollinators, honeybees and all that. So that really worked very well to us, and now after going a lot of ups and downs in seed production data, and now we are very confident where to grow hybrid seed, and now it's become very, very easy for us to identify. This year we have a lot of seed production program, and the plot setting, if we see and we have some judgment in our eyes, we can say the average we will get around 1.5 to 2 tons per hectare hybrid yield, that is A into R. It is very good. And another thing is happening in this case of Pigeonpea, seed weight is reduced to half. Now we are recommending 2 kilogram per acre. That means 5 kilos per hectare. That means very, very, very important that we grow at wide spacing, take a short intercrop, and also what we can do that we can really even the seed rate is 400 kg for 400 rupees kg. Farmers will not hesitate in buying it, looking at the productivity advantage. They have it. So these things work very well with us. Now you can see why this seed, why the pod set is good. And this is a picture from Patancheru, and you can see how much pod load is on the male style plant. Nobody will believe this kind of pod set, and thanks to bees and other insects here. And we can harvest from this kind of podding easily 2 tons per hectare. And that's what we have got repeatedly. Even this year, same field, RP16, we have similar kind of pod set on male style plants. So this is what our technology is all about. Male style plant which breed true, and also we have pollinators which can help us in producing hybrid seed. And once this is done, then there is no stopping for hybrids, right? So Madhya Pradesh we found the best state for hybrid seed production. Why? Because of some reason, and you can see here the yield we got. You can see the underlined one, 2.2 tons, 2.5 tons, 3 tons, this kind of yield we have got. If you look at the Tikam Ware, which is a real forest area, we got about 3 tons yield in 5 hectare blocks, huge block of seed production. That really gave us a lot of confidence that yes, now nobody can stop hybrid. It may be delayed, some problem may come, but our technology is perfect. Now, seed to production ratio, this is also very important for seed producer. If we grow 1 hectare of hybrid seed production, we can cultivate commercial crop in about 400 hectares. So that's very good for us. This is not happening in many crops, but PNP is very good. In fact, now private seed company people have started talking about it, that hybrid seed production in PNP is easier than castor. It is easier than many other crop because it's very simple. We don't recommend any special treatment to the crop. Only we need, it should be, sites should be selected where we have pollinators. That's what only recommendation. And now we have trained our staff, we have trained our seed growers, and this is not a problem anymore. Now, still we have to work on that how we can maintain quality of that, quality of purity of the hybrid, which is very, very important. Now, this character which you see, on this side you can see the leaf. This leaf is not normal. We call it obcordate leaf. And this character is controlled by single recessive gene. That means it can be transferred very easily to any line. And now we have done it. Last 4 years we were involved in that. Now our hybrids with this leaf are available. And once we make hybrid with the normal leaf, you can see what happens. The leaf is normal. What implication it has for seed purity? Very, very big actually, especially in multiplication of female parent. If you are multiplying female parent, and Pigeonpea, I must mention, there is no dormancy in the seed. And that means you can harvest and plant immediately. So what we recommend that those farmers who are multiplying female parent seed on large scale, they harvest their seed sample, grow anywhere in the pot or in the soil, and within 4 weeks they will know whether their seed material is pure or not. This is the cheapest technology, very reliable technology. So this character is getting a lot of attention from our group, and now we are transferring this character in very diverse line and also available to national program like that. So this is it, really very good step we have taken, and we have succeeded in doing that. Now for what we do immediately, quality again, I said very important. So we have taken help from Dr. Rajiv Varshini's group to help us, because it is very, very important for us, because Pigeonpea is a long duration crop. Number one. Number two, it is a photosensitive, as I mentioned. That means after harvesting hybrid seed, we cannot grow the crop up to flowering because it is photosensitive. It will flower only in short days. That means if we depend payment to farmers based on quality, we will lose 1 year. And no seed company would like to really compromise on this. So we have to do this kind of activity, and I'm very happy to mention that the Rajiv group had done a wonderful job by doing this very simple technology. You can see here in the bottom row, this is a male parent, you can see one peak, then middle row is a female, we have one peak, and what happens in hybrid, we have both the peaks. So this has implication that we can purify, we can test the quality of female parent, we can test the quality of male parent, we can test the quality of hybrid in very, very little time. And this kind of testing is more reliable than any field testing. I'm very happy to mention here that Rajiv is very considerate in giving this technology to anybody who wants who are involved in hybrid technology, and that's what we have to really look from ICRISAT, that we develop technology, pass on to the other who can use it. So this is what he has done, and now he has done same thing for other important hybrids, and this is very, very important steps. And Rajiv, I'm thankful to you for helping us in this. So we must not forget about genomics, which have helped us a lot. We want to continue with the genomic studies in our breeding program, and we think that this is a very important area. We should not be away from that. We should work together into this. And when we talk of specific traits, like I showed obcordate leaf, if we have really know that this particular gene we have to transfer, then I think job will be much easier. Otherwise, we will take 7-8 years to transfer that trait into a particular variety. But with the help of genomics, we can do it in no time, really. Then the limitation comes in genomics is that we cannot breed for adaptation. That means how variety will perform in farmer's field. So we have to work together. There is no option left to us. And I'm very happy again to thank Rajiv that he has understood this thing very well, that we have to work together, and we are working together, and we have a very strong team on working on hybrids and our variety work also. But we have to really rationalize that which character we have to take for genomic studies. We cannot work because resources are limited, people are limited, we can't do it for all the character. So we have to really sit together and work out, okay, we'll work on these traits only. So that way I think we can develop good populations. Now just few slides I want to show how this hybrid looks. This is in Andhra Pradesh, and look at this slide. I call it the Pigeonpea jungle in Maharashtra. This is a common scene this year because the rainfall was very good. And this plot was planted, row to row was about 8 feet. And you can see that it's very difficult to walk it between two rows, and you can see the plant is spread here, fantastic, and all plants are full of pots. This kind of thing can impress any farmer, any politician, any leader that okay, this is the type of thing we should have, and this year we are getting this kind of feedback from everywhere. Now, very difficult. question I have put here, that how we can achieve 1 lakh hectare which we have committed to Andhra Pradesh. So, Dr. Samir Kumar is in central stage. So if he doesn't achieve, where is he? So we can chop him. So we have worked very seriously onto this, how we can achieve 1 lakh hectare. And this year, looking at the seed production data, seed production fields, I have no hesitation that we will achieve that very easily. And in this, I must acknowledge the support of Andhra Pradesh government. They have really come forward, okay, we will help you all the way. So all the seed will be purchased by Andhra Pradesh government, and they will distribute with the subsidy to the farmers. And that's what we want. We cannot expect more than that from any government. And I can tell you at this time, Samir has played very important role in discussing with Andhra Pradesh government and negotiating this thing. So I'm sure next year he will give us good news that it is on 1 lakh hectare. Samir, you promise it? Yes. Yes, he is promising now. Now, in fact, baton I have given to my colleagues. Team will be led by Samir Kumar, followed by Vijay Kumar, Mayar and Anupama. They will, I'm sure, they will produce the results we want. Okay, Samir? So don't disappoint me. But we need lot of support from management. An important thing is positive frame of mind of management. They should really help us in doing that, especially monitoring, very, very important. Organizing meetings, they should provide us full support, and I'm sure Rajiv will not stand behind in that. He will come forward and help us the way we want to really monitoring, because without monitoring we cannot do, especially seed production. This year we have around 8,000 acres, am I right? 800 acres of seed production, and this year I think 90% of that has been monitored by Vijay Kumar and all the field staff and Samir. So we know from where to harvest seed, from where not to harvest seed. Okay. No problems we can solve, don't worry about that. Right. Now, this is very important slide as far as I am concerned. This, if you want hybrid to go, we have to transfer this technology free of cost to private seed company. That means, again, it requires very good discussion with management whether they can do it or not. At present, it is with the consortium, hybrid seed consortium, and this is not helping us at all. Because the seed producers are really thinking why to invest money so much while they are not earning money. That's the main thing. So I think for a few years we can leave this technology, because these two hybrids are already released. Let us help everybody to promote this hybrid and hybrid technology. Anyway, I leave this up to Rajiv to take up this material with Dr. Gowda and see what we can do it. Many private small seed companies want to come, but they are afraid to invest 6 lakhs per year in this venture. Now we have to, in future, we have to look for expenditure, not less than, say, we have to look about a very big target that I already told, and private seed company sector has to be with us. And this year we are really involving a lot of farmers and a lot of NGOs in the seed production, and we are ready for the big one now. Now we want to see that in 5 years we cover around 33% area, or 1/3 area of Pigeonpea. I hope we can achieve with all efforts from not only from ICRISAT but various state governments also. Because now we see positive trend in Maharashtra, the largest state which grows Pigeonpea are in our favor. Right. Then we have to really breed new varieties, environment specific varieties for future, and then we have to breed now. If you really want to break through in this further to breed hybrids which can produce 7 tons yield, we have to really go for environment specific hybrid. We cannot expect that every hybrid will do well everywhere. That's silly to really believe that. So we have to breed locally adapted hybrid for a specific environment. And I'm sure 7 ton is not too big, because I was telling 5 ton till yesterday, but now the farmers have reported 5 ton yields in their own field. We have not got that kind of yield at Patancheru, but farmers have reported 4.5 to 5 tons yield. And this year also we are going to get. So we should fix target little more, so that we are thinking about increasing yield at least in our breeding material, right? So this will try to do that now. The question is very specific, how we should get 7 ton hybrid. It is little scientific, but I'll not spend much time on that. We will make use of heterotic groups. We will prepare heterotic groups, and in this activity, the involvement of both breeders and genomic scientists are very, very important. Because what happens in Pigeonpea, whatever diversity we see, these heterotic groups are based on diversity. More diverse meteorology crops, we expect more hybrid vigor. But in Pigeonpea, it's a really funny crop. It looks very diverse, but once you analyze through genomics, diversity is very little. So we have to choose parents which are diverse at genetic level. That kind of parents we have to select and start using in the breeding program. So we have to establish those groups first. First we use the data of breeders so that we can identify broadly which are our heterotic groups, and then we go for genomic studies because genomics are expensive exercise, so we can do that. And then we start breeding. How we will breed that? Within a group, we will do intermating so that favorable alleles are put together, right? Then other step we will do it, we will start making crosses to identify which are good general combiner and good specific combiner. By specific combiner I mean two particular hybrids, when they come, parents will come together, yields are very high. But they cannot do this kind of thing with all the parents. So very specific combination we have to identify, and if we do it successfully, we will get very high yielding hybrid. And general combining ability is also very, very important, because this way we can reduce our breeding material which is not useful in it. So those lines which are good in, which combine well with everything, we call general combiner, good general combiner law. Now for getting this kind of yield, once we establish hybrid heterotic group, we have to make crosses between two diverse group. And I again say, diversity based on genomic studies. So we know these are really diverse, let us make it. We should not too much worry whether two parents are very poor in yielding. No, don't worry about it. Let us explore that. So that's why we have to work on it. So this is our future activity. And also, as I mentioned earlier, we have to start accumulating favorable allele from everywhere, wherever we get. And we have to take help again from genomics in this. So other areas other than hybrid is that we continue to breed very high yielding super early varieties which mature in around 85 days here at ICRISAT, and we already have this material, but yield is not so high. We will work on this, and we want to reduce our another losses which is caused by water logging. So we have got very good indication on that. Now we know that water logging resistance can be achieved by selecting genotypes which have lenticels, which produce adventitious root under waterlogged condition. So this kind of thing we have to put together in one genotype. And again, I will request Dr. Varshney to involve this character also in the genomic studies. Now we have parents identified. There are three criteria for water logging. One is they produce parenchyma cells that will hold oxygen in that inside the root, produce lenticels, and also produce adventitious root. So we have three different kind of parents identified. We can confirm this this in this particular year and start developing populations so that those can be categorized for genomic work. So now it is very, very important. We should go for new areas. Two examples we have set in last 4 years. One is promoting Pigeonpea, this variety ICPH 88039 in the hills of Himalayas. There we have got average yield of more than 1.2 tons per hectare. Very productive line. And you will not believe when I say that. Farmers have stopped growing rice in valleys and they say Pigeonpea we want to grow because Pigeonpea productivity is high in that system, investment is very little and market price is very good. And when we ask question, what about your rice? They said rice we can buy from market. And they are earning lot of money from Pigeonpea seed, from fodder, and more than that is the making basket from the Pigeonpea sticks. See, this is what happened in hills, because hills are producing lot of vegetable and fruits. So those people have already contacted these guys, so they are making basket and supplying. So this is this kind of additional income they are getting from this crop. Another example is how this variety has changed the life of farmers in Rajasthan. And Joanna has seen that, how it has changed really, and really farmers are very happy there to look at. I don't want to talk on that because it has been published in our ICRISAT happening quite a lot. And this is also, we have to look into that. This is again for our breeders. If we can incorporate Cryptogamy trace, self-link treatment, it becomes very easy to work it out. Now very little, very fast on what we are doing in Odisha. Dr. Meyer has put his whole effort in promoting Pigeonpea in Odisha very successfully, and is so successful now he thinks that he should settle down in Odisha. So now he's getting very good yields there, and now many people know Meyer there, right? Whether they understand his English or not, that's a different thing, but but he's getting his work done very effectively, right? And job is doing very well, so he's promoting. I think I will not talk much on that because this project is highly successful in the sense that Odisha government themselves have come forward with 2 new projects for Maya. And that's why they say they will give already citizenship of Odisha to him because he is helping poor so many times. Now, seed system again, the contribution from Dr. Marmula is immense. He is totally involved in this kind of system, and now he has become a big consultant in seed production. Even national program, people are asking how not to grow seed and how to grow seed like that. And again, I will request my Pigeonpea group here that don't shy away from publications, because until you publish, nobody will know you, you have to generate new information and always think of publications, right? Well, the question of USA, I thought somebody will ask questions, so I put this thing here. The hybrid program, definitely we have tried to introduce this year with the help of TL2, and the first training program was highly successful there, and I'm sure we will be able to do something there. But we need again priority, and this is a very good picture which we have taken this year. This is from Maharashtra, and in one field they have planted hybrid 2740 in 17 acres, and when I asked farmer, can you get 3 tons in this, you know what answer he gave? Sir, if it gives 3 tons, I will not grow this crop. That was his answer. I mean, he was so confident that he will get at least 3.5 to 4 tons from this. And on Friday he came here and he said, now I am very confident we are going to get not less than 4 tons per hectare. So this kind of thing, we need farmers like that, we need collaborators like that, and this is again done by one NGO in Maharashtra. They are organizing very well for us. Now, what farmers, our farmer want is like that. Okay. Now what we did, I think we can do it, but I am available. You can call me. See, just essential services, I am available on this number. You can call me anytime you want, only for hybrid, nothing else. Okay. Service is free, only transport will be required. Now, we have one gift, we call it departing gift, a new kind of thing we have got here, and that is the, we have recently got a male sterile line which is temperature sensitive, and myself and Dr. Ashwini's group is very actively working on that. In fact, he already identified exactly what temperature it changes. This is a male sterile which changes with the environment. That means if you grow, our field data show that if you grow more than 26 degrees, it is sterile. Less than 24 degrees, it is fertile. And the studies conducted by Dr. Vashni's group, we say 23 degrees is the critical point, like that. But we have to do a lot of work on that. We have to work on genomics. So I thought I will just share this information with all of you and treat it as my departing gift to ICRISAT, because it's a very recent one. Where are you? I want to, no. I want to show two videos, very small videos. Are you tired? Oh, that's good then. Then I'm sure you will enjoy what Dr. Elders, like Dr. Swaminathan talk about Pigeonpea hybrid, and then we'll show some other thing also.
Where you can put a legume. We can even today morning I saw Dr. Gowda and Dr. Saxena. Dr. Keating showed me this wonderful hybrid Pigeonpea developed here. It's going to really be the starting point of a pulses revolution in India. The hybrid Pigeonpea, I think it will be a trigger. Any revolution requires a trigger, like the semi-dwarf varieties of wheat and rice or hybrid corn became a trigger for a revolution. You require a major breakthrough, and I think the Pigeonpea will be very important. We have seen in our...
Now one more video is about me only. So here it is. And after that you can clap.
It's true of humanity. Tonight has been largely determined by social class. For the majority of people, the large portion of their daily diet is made up of grains. Grains are buried whole in seed, or seed, dried in flour, and made into bread. And even watered and grew as ever. All the time, there were hardly any legumes such as beans, peas, or lentils. Fish were available in hinterlands. Meat was meant for the land horse, and the poor could only get morsels on very rare occasions and festivities. Men and women both burned approximately 1,800 calories just by sleeping and walking. This, along with the calories required to complete a day's labor, must have been impossible to meet given the number of food sources. The history of Indian freedom struggle had indeed been a 200-year-old survival against hunger and epidemics. As a free nation, the growth of India has been remarkable in all fields, including agriculture, and yet, the scourge of malnutrition is not going away. The Prime Minister is honest and candid to call it as a national shame. A whopping 42% of Indian children are suffering from malnutrition, and the axis of the problem is mere absence of protein in their diets. Animal proteins have to be a part of poor people's diet, but how? In 1974, KB Saxena, a young graduate from GB University, hardly knew that he was starting a mission to grow protein for 1 billion poor. 4 decades later, KB Saxena stands tall in the community of agricultural scientists by developing the world's first hybrid legume. Making possible the widespread cultivation of pigeonpea, once considered an orphan crop and then be grown by small and marginal farmers across the world, he has now earned the crop a new honorific name, Poor People's Meat, for its protein-rich content. KB Saxena's work won him China's highest civilian honor, the 2001 Friendship Award, and a lead position in the National Food Security Mission of the Government of India. The story of KB Saxena is a story of innovation, perseverance, and selflessness, as much as the story of pigeonpea is the story of neglect, hardiness, and resilience. Both stories are intertwined as two strands of DNA links, linked by the base pillars of institutional excellence and individual brilliance, of scientific robustness and nature's adaptability. How come pigeonpea is loaded with protein? Pigeonpea comes from the fabaceae or legume family, the third largest family of flowering plants. The legume seeds develop from a simple carpel which usually opens around a seed on two sides. Growing legumes are cultivated for their seeds and include beans, lentils, legumes, peas, and chickpea. Legume plants are notable for their ability to fix atmospheric nitrogen through a symbiotic relationship with soil bacteria, rhizobia. By itself useless, once rhizobia enter inside root nodules of legumes, they fix nitrogen from the atmosphere into a plant usable form. In return, the plant supplies the bacteria with carbohydrates, proteins, and sufficient oxygen to proliferate. Pigeonpea is not new to India. The cultivation of pigeonpea goes back at least 3,500 years. The center of its origin is the eastern part of peninsular India. Pigeonpea naturally occurred in tropical deciduous forests, receiving annual rainfall of 1,000 to 2,000 mm, with distinct dry and rainy seasons and a small range of temperature. Pigeonpea is a drought-tolerant plant and can be grown in areas with less than 650 mm annual rainfall. Traditionally, the use of such inputs as fertilizers, irrigation and pesticides has been minimal. Today, pigeonpea is an important legume crop of rain-fed agriculture in the semi-arid tropics. World cultivation of pigeonpea is estimated at 46,000 square kilometers. About 42% of this is grown in India, followed by Myanmar and Nepal in Asia, in southern and eastern Africa, and Central America. But the yield levels of this crop have stagnated at a modest 700 kg per hectare. How to reach the 1 ton per hectare area? Can the yield of pigeonpea be taken to the levels of cereals? To achieve this target, a viable commercial hybrid technology was needed, similar to those available for open pollinated field crops and vegetables. But the self-pollinating nature of pigeonpea plant was standing in the way. The pigeonpea flower has both stamen and a carpel which contact each other, and the flowers self-pollinate as they are closing. Great pigeonpea flowers attract a number of insects, which affect cross-fertilization with an average natural outcrossing of up to 20%. Honeybee and bumblebees can carry 5,000 to 100,000 pollen grains, the male parts of the plant, on their body and effect cross-pollination. To develop a new variety, KB Saxena pollinated the delicate ovaries by hand, called Genetic Male Sterility System, or GMS. In 1991, ICRISAT released the world's first hybrid of a pulse crop, ICPH for commercial cultivation. The ICPH plants are vigorous, semi-spreading, and intermediate in growth habit, with profuse branching, flowering, and cutting characteristics, and prove outstanding in diverse environments, particularly in the central zone of India. The success of ICPH marks the beginning of a new era in pulse production. Despite demonstrating high yields, the crop cannot be upscaled because of high costs of hand pollination and the inherent constraints associated with seed multiplication of female parent line. Genes in a plant cell reside both in the nucleus and the gel-like substance between the cell membrane called cytoplasm. KB Saxena noted the genes present in the cytoplasm of a wild species with those present in the nucleus of the cultivated type, and their interaction conferred male sterility. As the cytoplasm is passed down through the eggs to all the progeny, the offspring of plants with a particular cytoplasmic genes would all be male sterile. It was called the Cytoplasmic Nuclear Male Sterility System, or CMS. How was it done? In both male sterile and fertile floral plants, cell division necessary for sexual reproduction, or meiosis, proceeded minimally until the chromosomes line up closely together as tetrads. When the parent mother cell wall dissolves, the tetrads card to migrate and fertilize. But if the wall is stopped from dissolving, tetrads remain trapped inside the pollen wall and make the flower male sterile. In the next 2 years, the pigeonpea team at ICRISAT produced a series of high-yielding hybrids and tested them at a number of locations. Among the short duration hybrids, ICPH 2433 recorded the highest yield of 2.4 tons per hectare. ICPH 2438 produced 4.5 tons per hectare yield at Aurangabad in Maharashtra and 3.4 tons per hectare in Nizamabad in Andhra Pradesh. For a crop that never saw 1 ton per hectare yield, it was truly incredible. The focus was now shifted to resistance to diseases. CNS line ICPH2043 was developed by crossing known disease-resistant restorer lines. The hybrids ICPH2671 and ICPH2740 gave yield gains of more than 30% without any disease incidence. ICRISAT's Pigeonpea variety ICP8863 revived the crop in central India, especially in Andhra Pradesh, Karnataka and Maharashtra, where the Turkey roots disease had devastated all previous plantings. It was called Maruti by the grateful farmers, revering Hindu god Hanuman, the destroyer of all demonic forces. The visionary Director General of ICRISAT and votary of science with a human face, Dr. Legumes, backed the development with all needed resources and unshaken belief, and turned the achievement epoch-making in the history of plant breeding. Dr. declared that the hybrid technology will benefit not only Indian pigeonpea farmers, but also those in Africa and Myanmar. The Indian Council of Agricultural Research, ICAR, participated wholeheartedly and provided generous financial support. To sustain the hybrid technology on a long-term basis, the next step was to involve private seed companies. Hence, C.L. Research Program Director on Grain Legumes at ICRISAT, initiated several seed companies into the hybrid pigeonpea seed business. In the farmers' fields, the hybrids outshone predictions. In more than 2,000 on-farm trials conducted in the states of Andhra Pradesh, Karnataka, Maharashtra, Madhya Pradesh and Jharkhand, the ICPH2671 demonstrated 14% yield advantage over best local varieties. The hybrid acquired the name Pushkar, the Sanskrit word for abundant and full. Under hybrid systems, ICPH2740 has even produced yields close to 5 tons per hectare in farmers' fields, a world record for any pigeonpea cultivar. ICRISAT believes that the CNS-based hybrid pigeonpea technology is ready for delivery. Sometimes a fine-tuning of the seed production technology may be needed to suit different environments. Pigeonpea is predominantly cultivated by small-holding farmers, and it is imperative that the seed costs remain within the reach of resource-poor growers. At present, both ICRISAT and ICAR are actively involved in the crop's technological improvement and its transfer to various developmental agencies. The Ministry of Agriculture in Uttarakhand has now decided to extend pigeonpea cultivation on 100,000 hectares in 9 hilly districts under the leadership of KB Saxena. This endeavor has been targeted with objectives of controlling soil erosion and enhancing nutritional and financial security of the poor farmers. Pigeonpea is no more an orphan crop, but part of the national food security mission in India. The dream of a young plant breeder indeed came true. India's pigeonpea production accounts for over 70% of the global and 80% of Asia's total pigeonpea production. There is a demand and supply imbalance. India is set to increase its pigeonpea production from 2.3 million tons in 2010 to 5 million tons in 2020. increase in production will come mainly from yield improvements, as only a marginal increase in area is possible. So, what is next? Recently, a global team of scientists led by ICRISAT completed and published the genome sequencing of the Pigeonpea genotype ICPH 7119, aptly called Asha, word for hope, in India. If Pigeonpea was to be a novel, it could potentially have 48,680 characters or genes. This is the first draft genome sequence for a growing legume. A couple of hundreds of these genes were found unique to the crop in terms of drought tolerance and important trait that can be transferred to other similar crops like soybean, cowpea, or common bean that belong to the same family. In the long term, with the help of low-cost sequencing technologies and approaches, the draft genome sequence will facilitate understanding of the genetic basis of many traits of the crop at the genome level. KB Saxena has truly developed Pigeonpea across the Rubicon. Pigeonpea cultivation has passed a point of no return. Let there be no recoil for want of hybrid seed. Let us work not only to ensure that India need not import any Pigeonpea, but also that this poor-man's protein source grow throughout the semi-arid tropics of the world. It was not merely a matter of hard work, but a creative and unwearied mind that wanted the Pigeonpea research that counted. Let us salute the team of agriculture scientists, farmers, and officials who are not content to accept the status quo and settle with small things. Let us not leave great things undone. Let every grower receive his share of protein and grow to be productive and perfect.
So, thank you very much for your patience, but you have seen what Pigeonpea has done to me, right? So, now I conclude my presentations, and if there are any questions, we can take care now or in the evening, no problem.
Thank you very much, sir. But once again, please join me in thanking Dr. Saxena for wonderful presentation. And I think I would like to say a few words very quickly. Please take a seat here. So first, this Dr. Saxena, when he made the presentation, he shared his work, research experience at ICRISAT, what he has been doing for last 40 years. And because of hard work of him and his team, of course, several things came out very clearly that because of his hard work, that Pigeonpea became the first crop in many instances. For instance, we had the first GMS in Pigeonpea, we had first cytoplasmic male sterility in Pigeonpea, we had the first time hybrid system in any legume crop in Pigeonpea, and of course, maybe I'm a little bit biased, but if you see the genomics result, then even in that context, Pigeonpea became the first crop where ICRISAT or any CGR system led the genome sequencing. And because of all these things, again, this is the hard work and of Dr. Saxena and his team, that ICRISAT has become come on the international map on the different aspect that Pigeonpea is doing this. His presentation is also an inspiration, in my opinion, for many of us, for the young generation, that there was a time, and if you see his presentation, you can see two different phases. One is probably 1974 to 96, where they did a lot of work in the area of varietal development, etc. And then this was 96, that even Pigeonpea became out of the mandate crop. But he did not lose his hopes, he did not lose his hard work, he did not stop. And he made it sure that because of his hard work only that you had the CMS. And this is something that we need to talk, that high quality science. If we didn't have those CMS, maybe Pigeonpea was not a part of the ICRISAT mandate crop. But he did not lose that kind of thing, and he continued to do or deliver outstanding quality science product that they made it further, or they made it sure that one Pigeonpea remains to be the ICRISAT mandate crop. And as a result, you can see that in the second phase, we delivered much more. Our Director General and always talks about I mode. We always say that farmers, they should be in the center. Again, hybrid technology is one of those technology which are providing access to the farmers to the market. Now, Jonna and her group, we are developing those I mode exemplar, working with Cynthia, etc. Here, this is very clear that now the Rajasthan case, what you demonstrated, that how this hybrid technology is helping farmers to come out from this poverty trap and then taking them towards the prosperity. Last week, I was in ESA and Eastern Southern Africa, where we had this regional planning meeting. I visited the field, where again this is very important, that ICRISAT is not just working in Asia. We are global. We have global mandate. We need to transfer those technology in Africa, and this was a great. I was very happy to see that we are working in this direction. That basically that how to convert African lines or how to adopt the cytoplasmic meal stilate in Africa, and I was very much pleased to see that now our colleagues have already started to work in this direction. So now, as a research program director, this is also my responsibility, and I'm telling my colleagues there that guys, let's have this hybrid technology in Africa, and this is going to be very, very helpful. Colleagues from Asia like Sami Kumar and others, they will be helping. This is one of the reasons that in the month of December, and I'm talking not only for ICRISAT based in Nairobi, but we organized a training course in December where Samir, Vijay Kumar, Antama and many colleagues went from there. In that meeting, in that training, we had national partners from East and Southern Africa. And while talking with some people like Egerton University, Paul Kimeto and many other people who participated in this training, I'm talking NARS partner, they were also excited. They say, we need to have this hybrid technology here. So you can see that, well, the work what Dr. Saxena has done, that not only restricted to Asia or these things, but to outside. As you mentioned again, that even this was the first crop where private sector gave the contribution. So again, in the end, hard work pays, and we as young generation, etc., we should always keep this thing in our mind. And now I do not have any intention to summarize his presentation because this was very clear, and then we could also see, and this is thanks to you that you also showed us a video which mentions very nice word, a nice work about you, about ICRISAT, and we are very much pleased about that. And I think if we would like to have one or two quick questions, we can have here. Otherwise, as Dr. Saxena mentioned, that he is available for essential services, and these questions may be part of those essential services. If not, then we can also, well, we can have just one quick, some queries, etc. Or this was good that you also gave the words of wisdom. You mentioned many things, and I have taken note. We'll be discussing with my colleagues and also to the management. And of course, we know that you are leaving big shoes. It's really difficult to fit in that, but we will do our best. And as you mentioned, we are thankful to you that you mentioned that although you are superannuating from ICRISAT, but your services not from hybrid and you will be available when, as and when we need your help. So we really appreciate that. So any question, any query right now? Yes, Rajesh, please.
I'm conscious of the time, but just one quick observation is to say we cannot congratulate you enough for your tremendous and great achievement over the 4 decades. But very quickly, if you have to reflect back, what would you have done differently? Because there's an implication for the entire breeding program going forward as to, if you were to go back and restart this, would have been done shorter period, could be things have been differently? Very briefly, if you would like to say something.
Thank you. I think this is very relevant today. And I would like to mention what we really did not do in 1974 to 96, which would have made a picture very different. And one thing we missed that time, and I was discussing this with my colleague in car also, is that we did not give enough emphasis for promoting PNP in Africa. That was our first mistake. I wouldn't say mistake, but that was perhaps the decision of the management that time, we honor that. That time there was no money drawn from, and we could have done it very easily. Second part is that we did not really give attention to promoting PNP in Central America. Right, there also PNP is very important. Farmers are very poor there, and they mainly depend on selling vegetable PNP because dal is not a commercialized product there. So these kind of things we did not really put a lot of effort that time, and we could not make it a global crop that time. And when time came in 1996, even our board and all said, oh, it is Indian crop. So that kind of very important thing we missed in the early stages. Maybe that was the right decision at that time, but we think now that we could have done much better making it an international crop that time because funding was not a problem. Alright, sir. Thank you. And in the end, I would like to acknowledge my team and request all PNP scientists to get up and show their faith that everybody has contributed, not only me. So please can I request all PNP. Sorry, starting. Everybody, yes, come on. Come on, these are my favorites. Yes. So they have done a wonderful job and will continue working for us. Thank you very much. You know which people working in Pigeonpea, they should also stand up. I was telling that. I was going to tell them to thank you, I think that's what leadership is all about. These people are not taking part of the UC. Okay, thank you very much. Yeah, last thing probably that we at ICRISAT we always talk about inclusiveness, and his presentation also gave very clear idea in the phase 1, of course together with Dr. Graham and Dr. Saxena. The Pigeonpea breed was just not solitary. They started to collaborate with the physiologists, with the pathologists, and even now, I will tell that story in the evening because I don't want to tell each and everything. So even when I came in 2006, Dr. Saxena was not talking at all about the genomics, even he would like to meet those folks. And I was very much happy to say this thing that now, in this presentation, where he summarizing the work for 40 years, genomics was coming very frequently from his, not just for the sake that what he needs to, but you could say, this is another thing that I'm talking from this global perspective. So genomics or any technology, we should not work just for the sake of technology that we see equals genome, we did this thing. The very important point is that when we are making the use of those technology, now Dr. Saxena mentioned, even hybrid technology, now how markers can be used. We are talking about open access. When we were talking in any crop, let's say, this hybrid purity, you should have some free, etc. So now we believe in this open access. Let's transfer this thing. Now the new areas which are very challenging. He mentioned that now this new areas, heterotic pole or this thermosensitive line, etc. So now colleagues from and here we believe in the team. So right now we are having this group genomics, breeding, pathology, entomology, etc. just for the management purpose. But we are a ICRISAT team. So when we are working on Pigeonpea, etc., we need to bring people from the different disciplines. So once again, and so now you can see that he has brought genomics so integrated in breeding program that we feel very proud of that, and by doing this thing at international level also. So now even the Plant Animal Genome Conference, which is the world's biggest conference in genomics, every year now we go there, and now even this year many people asked, Rajiv, this is really surprising. 4 or 5 years back, nobody knew about ICRISAT in genomics area, nobody knew about this chickpea, Pigeonpea, etc. Now you guys are having so many presentations, invited presentations, and everyone talks about that, and this gives us really proud that well, as a ICRISAT team, we are making something. With these things, I think we can conclude the seminar, and our sincere thanks once again to Dr. Saxena for all your hard work. And Rajesh Agrawal as OICDG already congratulated for all your work, and then we will be having him in the afternoon when we have the farewell session. And all of you are invited to join us for farewell of Dr. Saxena. This will be starting at 3 o'clock? 2:55, okay, 2:55. Thank you very much for coming here and have a nice day. Thank you very much. Very good.
Thank you very much. Thank you for coming.